Hello dear learners, today we are going to discuss the preparation, that is simple syrup ip66. Now why it is ip66? Because the preparation's formula is official in Indian Pharmacopoeia 1966 edition, so it is being named as simple syrup ip66 and the formulation is from the pharmaceutics lab. Okay. Aim of the experiment is preparation and evaluation of simple syrup ip66. That is, first we are supposed to prepare and then we are supposed to evaluate that syrup. So for that, the preparation requirements are, first is beaker, as you can see here in the image, it is beaker, then glass rod, okay, then water bath is there. So these are the water baths which are used generally in the lab. This is the conventional one. Then tripod stand is there. Glass bottles are there for the storage of the syrup. Two bottles you can see here. One is transparent bottle and the another is amber color bottle, just for the protection purpose from the light if light sensitive drugs are there. So these are the requirements. Next we will see the formula. So formula says that as the name simple syrup, it is having only two ingredients. One is sucrose and the another one is purified water. So the sucrose, according to the Indian Pharmacopoeia formula, the quantity is 667 gram and the purified water is quantity sufficient to 1000 gram. So this 667 gram according to the Indian Pharmacopoeia is the quantity given. Now we are supposed to calculate the quantity taken for required quantity of the preparation. Like if you want to prepare 15 gram, if someone wants to prepare 20 gram, so it will vary according to the quantity. So here we have taken 15 gram. So for 15 gram it is how much? So for that you are supposed to calculate it like this way. For 1000 ml it is 667, as you can see here, for 1000 it is 667 gram of sucrose. Then for 15 it is how much? So simply we are supposed to cross multiply 15 into 667 divided by 1000. So it will give you 10.005 gram of sucrose, which is to be added to the preparation. And the categories, as you can see here, the activity of the ingredients, the sucrose is acting as a sweetener which is imparting the sweetening properties to the preparation and purified water which is used as a vehicle here. Okay. So as you can see in the image, it is sucrose formula, which is a disaccharide which can be split up into two monosaccharides, that is glucose and fructose after hydrolysis. These are the two processes wherein we can obtain the water. Now this is the distillation unit for obtaining the distilled water and this is the purifier for obtaining the purified water. Now why purified water? Why not distilled water? The reason is, first is purified water undergoes a different series of filtration processes which are quite simple, and the processes are like deionization, reverse osmosis, carbon filtration, etc. And the purified water is having the necessary minerals into it, whereas distilled water is completely devoid of the purities and the minerals. And distillation process is bit tedious process and it is long process also, since the boiling water is collected and the steam, means boiling water and collecting the steam that forms in the container. Since the components of water have different boiling points, the process is carried out for multiple times. So since it is carried out multiple times, it is long procedure and it is tedious also, and it is devoid of minerals and purities as well. So the reason is purified water is preferred for the pharmaceutical preparations. Next is procedure. The procedure says, as we have seen the requirements, we need beaker, we need glass rods, okay, then we need water bath and all. So first we are supposed to take, as you can see here in the picture, we are supposed to take required quantity of sucrose, and then 1 beaker we are supposed to take, and glass rod. So you are supposed to place them, the beaker and the glass rod, on electronic balance first, and tearing is to be done. Now what do you mean by tearing? To make it auto zero, like the weight can be subtracted of that of beaker and the glass rod from the weight of the preparation. So required quantity of sucrose is weighed, like 10 gram we are supposed to take for this preparation to make it 15 gram, and this is transferred to the tiered beaker, and sufficient quantity of water is being added to the beaker, and it is placed on the burner. It is heated and stirred with glass rod continuously till the sucrose dissolves. Like till the sucrose completely dissolves, we are supposed to stir it continuously and also continue the supply of heat. And the beaker was cooled to room temperature. Once the beaker comes to room temperature, we are supposed to add again the required quantity of water to make it to the required weight, that is 15 gram, and we are supposed to filter it and transfer it to the glass bottle. Okay, so these are the steps, or these are the procedural steps, and the method which is followed for the preparation of syrup is, it is with the aid of heat. That means you are not supposed to simply dissolve sugar in the water, you are supposed to dissolve that sugar in the water with the aid of heat. Next part is category and uses. The preparation category and uses are, it is first acting as a pharmaceutical aid. Now since it is aid, that means it is helping, and pharmaceutical, that means to some pharmaceuticals. So how the pharmaceutical aids are nothing but these are the substances which have little or no therapeutic value, but they are essentially used in the manufacturing and compounding of various pharmaceuticals. They don't have any therapeutic value of their own, but just they are helping in compounding of different pharmaceutical preparations. Like for example, medicated syrups are being prepared, and so that time these can be used as bases. Okay, then sweetening agent, next category is sweetening agent. For sweetening agent, since it contains sucrose which provides the sweet taste to the food, for example honey hai, okay, artificial sweetener saccharine is there, and this preparation is also being used as the base for preparing various types of syrups like medicated syrups, flavored syrups, etc. Next is the theory behind the syrup formation. Now according to the definition, it is 66.7% weight by weight of sucrose which is there in the formulation. So it is called as the concentrated aqueous preparation, concentrated. Since the sucrose percentage is 66.7% weight by weight, that is nothing but it is 67 gram approximately to 100 gram. Matlab agar 100 ml water if you are taking, then the amount of sucrose to be added to that 100 ml is approximately 67 gram. So you can imagine the extent of the sugar, like how much concentrated preparation it is, and it is dissolved in water, so it is aqueous preparation. And since the concentration of sucrose or sugar is more, it is concentrated, so it is concentrated aqueous preparation of sugar or its substitute. It can also be with flavoring agent or without it, and it can also be with some medicinal substance or without it. So when it is with any medicinal substance, it is called as medicated syrup, and when the syrup is with some flavorants, it is called as flavored syrups. For example, cherry syrup is there, okay, cocoa syrup is there, and the medicated syrups are like it is corexanol, okay. So these are the syrups which are medicated, that means they contain the active medicament. Then the syrups usually contains sweetening agent and viscosity builder Like as you can see in the preparation, sugar is not only imparting the sweetening properties, but it is also imparting viscosity to the preparation. Then antimicrobial preservatives can also be used, flavorants can also be used, colorants can also be used for the preparation of syrup. Then preservatives which are to be used in various medicated syrups and other syrups except simple syrup are benzoic acid is also there, sodium benzoate is there, parabens are there, methyl propyl parabens are there, alcohol is there, and the different concentrations of preservative are used to preserve the syrups, and they play different roles. They have different mechanism of actions. Like for example, if you will consider benzoic acid, how they act as preservative, they disrupt the cell membrane function और they inhibit the enzymes. Like बैक्टीरिया के सेल मेम्ब्रेन का जो फंक्शन है उनको डिस्टर्ब करना सेल मेम्ब्रेन के, या फिर एंजाइम्स को इनहिबिट करना इज द mechanism of benzoic acid. Next is simple syrup. As we discussed, it is 66.7% weight by weight of sucrose, and it is to be stored under cool storage conditions, because when it is stored under cool storage conditions, some sucrose may crystallize out from the solution when the syrup is completely saturated with the sucrose. Since it is saturated solution of sugar, some sucrose may crystallize out, and this crystallization means if the temperature is too down, the crystallization can occur. So to prevent this crystallization, the sorbitol can be added to prevent it, and this will prevent the recrystallization of the excess sugar from the supersaturated sugar solution during the normal storage conditions. And even if the temperature is too warm, then it may lead to the problems like fermentation, and if the temperature is too cool, then it may lead to the problems like crystallization. Okay, so as you can see here in the diagram, the crystallization is nothing but it is getting converted to the solid form, like sugar which is getting crystallizing out, and in fermentation here you can see the sugar and the yeast is getting converted to the alcohol and the carbon dioxide. See here you can see this is the fermented syrup. Since the CO2 is evolved, this process is fermentation, so which is not desirable. So if we are storing it at too warm temperatures, then this problem may arise, and too cool storage, this crystallization issue may arise. Then preparation we have just now discussed. There are various methods with the aid of heat, without the aid of heat. Okay, then addition of sucrose to a prepared medicated liquid or flavored liquid, that means medicated syrup preparations using simple syrups, and so on. So these are various methods, but here we are using the solution with the aid of heat, which we have already discussed that the heat stable components can be added to the hot syrup. Okay, so since the heat is involved, the heat causes the problems. Like if proper heating is not done, if this precaution is not taken, then the excessive heating which occurs, it may lead to the hydrolysis of sucrose into dextrose and levulose, or you may call it as the sucrose may be hydrolyzed to glucose and fructose. It is one and the same. Why? Because dextrose is what? It is a naturally occurring dextrorotatory form of glucose, and fructose is fructose, if you will see the levulose, as the name levulose, it is the levorotatory form of fructose. So this is nothing but same, and this reaction of getting sucrose getting hydrolyzed into glucose and fructose or dextrose and levulose is called as inversion, and the combination of two monosaccharide is known as invert sugar. So this inversion phenomenon is also important from the synopsis question point of view. Okay, as you can see here in the image, invert syrup formation, like sucrose which is getting converted into glucose and fructose, that is dextrose and levulose. Okay, this is inverted sugar, this is normal sucrose which is getting converted to glucose and fructose. Okay, next is the solution with aid of heat is what? It is heat is being utilized, or heat is being applied to the preparation of the syrup, and this some inversion which is occurring of that of sucrose is greatly influenced by the presence of acids also. Okay, so these acids are an unintended part of the preparation. Then invert sugar which is formed is more sweet than the actual sucrose, and the syrup darkens due to the overheating of the fructose, and this overheating it becomes like amber colored ka preparation, which is nothing but caramelization. When this syrup is overheated, it becomes amber colored, like as we have seen in that type of bottle, and the caramelization of sucrose occurs. Now here it's the question answer time. Be curious, like Albert Einstein says, I have no special talent, but I am only passionately curious. So the question answer session says, why syrups act as a self preservative? Like simple syrup here we are talking of. Now there are two things: the sucrose which is getting partially hydrolyzed to the glucose and fructose, or dextrose and levulose, it acts as antioxidant. So it is protecting the preparation, it is preventing the oxidation of the preparation. And the second reason is, since it contains high amount of sugar, okay, since it is nearly saturated solution, it is having a great osmotic pressure, and it is having the capability of water withdrawal from the microorganism, which leads to the dehydration of microorganism, and in turn microorganisms can't grow. So because of these two things, the syrup is having self preservative action, simple syrup. And the fermentation process which we discussed, that the evolution of alcohol and carbon dioxide, if the container is very tightly packed, then there are chances of bursting. So how you can prevent it? First thing, the container can be loosely loosely packed. Okay, the stopper which is there, it should not be very tightly closed. Then warm places should be avoided, which is giving rise to the evolution of carbon dioxide. Dry bottles are to be used to avoid the moisture. This moisture is also an unintended thing in the preparation of syrups. Okay, because the moisture, if it is there, then it is diluting the preparation again. Then next is store in a closely stoppered bottle. क्लोजली स्टॉपर्ड बॉटल में स्टोर करना है, सो दैट द unintended things can be avoided. And cool syrup to be filled in the bottle at room temperature. Since hot, if we are pouring hot syrup in the container and we are closing that container, then again the fermentation because of the warm water can give rise. So we can avoid this by all these things, avoiding the warm places. Then cool syrup is to be filled in the final container. Then closely stoppered bottle में store करना है after once it is being properly cooled. Dry bottles use करने हैं. Okay, so these are the ways by which we can reduce the fermentation of the sugar syrups. Then cool temperature storage, like why? Because warm temperature, as we discussed, it leads to fermentation. Okay, and the very cold temperature can lead to crystallization. So to avoid both of these, we are storing it at proper storage, that is cool temperature. What are precautions? Very first is small quantity of water is to be added for the sucrose dissolution, not whole quantity. Heating should be proper. Why? Because if heating exceeds, then fermentation of caramel can occur. Then storage, filtration, cooling, all the parameters should be proper. Then next, organoleptic evaluations are there. So these are color, odor, taste, clarity, pH, viscosity, weight and specific gravity. We are supposed to observe the color of the preparation, odor, then taste. It is having sweet taste, definitely. It is having a clear appearance, acidic pH, viscosity, weight and specific gravity. These are also the required parameters which are to be tested. Label, as you can see here in the diagram, the right hand side at the corner, top corner, it contains the quantity, and at the front it is having the name simple syrup IP66, and your right hand side, if you will see, all the manufacturing details are there. License number is there, batch number is there, manufacturing date is there, expiry date is there, then MRP is there and manufactured by whom. Okay, and your left hand side, if you will see, the content is mentioned. Now this content is nothing, each ml is not there. It is since it is a pharmaceutical aid, we are supposed to mention it in percentage. Okay, so this percentage content जो होता है sucrose IP, it is, since here I have written each ml, that is 0.0667 gram, but since it is a pharmaceutical aid, you are supposed to write it in the percentage, that is it is 66.7% weight by weight, and each ml you are not supposed to mention since it is a pharmaceutical aid. Then so intentionally I have kept it like this to tell you all that it is 66.7% you are supposed to mention when it is the pharmaceutical aid preparation. Category is pharmaceutical aid and sweetening agent. Storage, we have seen that it is to be stored in cool dry place, and we are supposed to replace the cap tightly after use. And at the bottom you find an instruction, that is it is not an instruction, it is just a manufacturing detail again, that it is manufactured at which place. And these are few of the questions, like whatever the things in video we discussed, it is like what is syrup, okay, then pharmaceutical applications kya hai syrups ke, why they are acting as self preservative, okay, 2 reasons we have seen, then why syrups at fairly cool temperature we need to store, then how fermentation can be reduced, which we have seen, what is inversion we have seen, fermentation, crystallization, all these parameters we discussed. Then what concentration of sucrose is suitable for the syrup preparation, then name and give the concentration of the preservatives used in the preservation of syrup, which also we discussed. Then factors which are affecting fermentation is temperature and moisture, okay, which we discussed. Then additional preservatives, if they are required in syrup, why they are required. Generally they are required in case of medicated syrup, not in simple syrup, but these are the possible questions from the syrup part. So with this we finish with the simple syrup Indian Pharmacopoeia 1966 formula and all the details like procedure and all. So for more details you can visit for more videos to our YouTube channel, Instagram, Facebook page, okay, and the site, that is Pharmacy Infoline, www.pharmacyinfoline.com. With this we finish with today's topic, that is from the pharmaceutical lab, that is simple syrup IP66. Stay tuned for the next preparations from the pharmaceutical lab. Thank you.